All right, let's jump right in. Today, we're going to be doing a deep dive into red blood cells.
Oh, cool.
You probably think, oh, I know all about red blood cells, they carry oxygen, but there's so much more to it.
Right.
We've got this physiology handout from Dr. Rasha Eldeeb,
Okay.
and we're going to uncover some really surprising details about their structure and their function,
Yeah.
even how they're made.
Wow.
So get ready, this is going to be a wild ride.
Okay, let's do it.
Okay, so starting with the basics, red blood cells, they're essentially sacs of hemoglobin, but they've got this really unique shape.
Yeah, it's pretty wild.
It's called biconcave. Have you ever heard of that?
I have, yeah. It's like a donut with a pushed-in hole on both sides.
Exactly. It's like a donut where someone took their thumbs and pushed in from both sides.
Hey.
And so that shape isn't just like a random quirk, right?
No, not at all.
And it actually plays a really important role in their function.
It's crucial for how they work.
And it's all thanks to these proteins.
Yeah.
Like ankyrin and spectrin.
Yeah. Oh, yeah, I've heard of those.
That basically create a scaffold inside the cell.
It's like their own little internal skeleton, keeping them structured.
Right, exactly. It gives them that structure, it holds that unique shape.
Yeah, yeah.
And the handout uses this really interesting word to describe the membrane. It says it's plastic.
Plastic. That's a weird word for it.
I know, right?
But I guess in a way it makes sense.
What do they mean by that?
Well, think about how these cells have to squeeze through those tiny capillaries, right? Like the teeny tiny blood vessels.
Well, yeah, like the tiniest ones.
So that flexibility, that plasticity, it lets them, bend and contort.
Oh, so they can squeeze through.
Exactly. They can get through those tight spots.
Wow.
That's pretty impressive when you think about it.
Yeah, that's amazing. Okay, so we've got the shape, we've got the membrane.
Right.
But what about the hemoglobin inside?
Yes. The good stuff.
I mean, we all know it carries oxygen.
Right.
But what else is going on in there?
Oh, there's so much more to hemoglobin than just oxygen.
Really? Like what?
Well, one thing that people don't often realize is that it plays a huge role in pH balance.
pH balance, like how acidic or basic something is.
Exactly.
But how does hemoglobin do that?
So it acts as a buffer, helping to keep our body's pH level stable.
Oh, wow. I never really thought about that.
Yeah, think about it. You can chug a big glass of acidic orange juice, and your body's pH doesn't go haywire.
That's true.
Hemoglobin is a big reason for
That's
that.
sort of built-in pH regulator.
Exactly.
That's incredible.
And of course, we can't forget about the membrane's other crucial role.
Right.
It keeps all that hemoglobin contained.
Why is that important?
If it were to leak out into the bloodstream,
Oh.
that could cause some serious problems.
Yeah, I guess that makes sense.
It's like a guardian making sure the hemoglobin stays put.
Okay, so we've got pH balance, we've got hemoglobin containment.
Uh-huh.
But wait, there's more.
Oh, yeah. Always more.
The membrane also plays a role in something else, something really interesting.
Okay, tell me.
You know our blood types, the whole A-B-A-B-O system.
Yeah.
Well, the membrane is involved in determining that.
Wait, really?
Yep, through this layer of glycoproteins.
Wow, so much going on in that little membrane.
I know, right? It's amazing how much responsibility these little cells have.
It really is.
And speaking of responsibility, they're involved in something else too.
No, what's that?
Blood viscosity.
Blood viscosity.
You know how thick our blood is.
Oh, right?
And that actually plays a role in maintaining our diastolic blood pressure.
Oh, that's interesting.
So it's like...
It's got to be the right consistency, kind of like
Yeah.
like making a sauce,
Okay.
Can't be too thin or too thick. It's got to be just right to coat the pasta properly.
So the pasta is our circulatory system.
Exactly. And the sauce is our blood.
That's a great analogy.
Thanks.
Okay, so we've got oxygen transport, pH balance, blood viscosity, and blood type determination all packed into these tiny little cells.
Pretty mind-blowing, right?
It really is. But how are they made?
Ah, the million-dollar question.
Where does it all begin?
Well, the bone marrow is like the red blood cell factory.
Oh, okay.
But like any good factory, it needs the right raw materials.
So what are those?
Well, you need proteins for the globin part of hemoglobin, iron for the heme part, and then a little bit of copper and cobalt. They act as cofactors.
Oh, okay. And don't forget about those essential vitamins.
Yes, absolutely. Especially B12 and folic acid, they're crucial.
So those are like the fuel that keep the factory running.
That's a great way to put it.
But there's another organ involved that might surprise you.
Oh,
What's that?
the liver.
The liver. I thought that was mainly about detoxification.
It is, but it actually has a hand in red blood cell production too.
Huh. Interesting.
So it produces the globin part of hemoglobin.
Okay.
It also stores some vital nutrients like vitamin B12 and iron.
So it's like the warehouse for the factory.
Exactly. And here's a key function. The liver secretes erythropoietin.
Erythropoietin. It's a hormone that stimulates red blood cell production.
Oh, so the liver is like the foreman, making sure the factory has all the supplies it needs.
That's a great way to think about it.
Cool.
But how does our body know when to ramp up production?
Good question. Our bodies have this amazing feedback system.
Okay.
So when our oxygen levels drop, a condition known as hypoxia, the kidneys and the liver release erythropoietin.
Ooh, okay.
And that signals the bone marrow to make more red blood cells.
So it's like a built-in sensor, always monitoring and adjusting.
Exactly. It keeps everything running smoothly.
That's incredible. So we've talked about where they're made, but I'm curious about hemoglobin itself.
Oh, yeah. Hemoglobin is fascinating.
What can you tell us about its structure?
Well, picture a globular protein
Okay.
composed of 4 subunits,
4.
each containing a heme group with an iron atom and a globin polypeptide chain.
So it's kind of like a 4-leaf clover.
Yeah, I like that. Each leaf being a subunit.
Okay.
And that iron in the heme is what binds to oxygen.
Right, right. And those globin chains, are they all the same?
Not all identical. There are actually different types, which leads to variations in hemoglobin.
Oh, right.
But we can dive into that another time. For now, what's really cool is how hemoglobin interacts with different molecules.
Okay, let's talk about that. We've talked about oxygen binding, but what else is there?
Well, one interesting interaction is with 2,7,3-diphosphoglycerate.
2,7,3-diphosphate what?
2,7,3-diphosphoglycerate. Or 2,7,3-diphosphoglycerate.
Okay, 2,7,3-diphosphoglycerate.
Think of it as a bit of a party crasher, competing with oxygen for those binding spots on hemoglobin.
Oh, so it's like a tug of war between oxygen and this 2,7,3-DPG.
Exactly. And who wins that tug of war is influenced by things like pH and temperature.
Wow, so it's all very dynamic.
Very much so.
Okay, so we've got oxygen and 2,7,3-DPG. But are there any other molecules that can interfere with hemoglobin's work?
Oh, for sure. And one that's particularly dangerous is carbon monoxide.
Carbon monoxide.
Yep. It binds to hemoglobin way more strongly than oxygen, basically blocking oxygen from getting on board.
So it's like oxygen can't even get a seat at the table.
Exactly. That's why carbon monoxide poisoning is so serious. It essentially starves your body of oxygen.
That's a chilling reminder of how delicate these systems can be.
Absolutely.
Yeah.
It makes you appreciate those everyday processes that we take for granted.
It really does. But what happens to hemoglobin when it reaches the end of its life cycle?
Well, it gets broken down
Okay.
into its hemoglobin components.
Right.
The globin is recycled back into the protein pool.
So it's like a circular economy.
Exactly. Nothing goes to waste.
Yeah.
And the heme gets further degraded.
Wow, we've covered so much ground already. From the unique structure of red blood cells to the intricate functions of hemoglobin, it's clear that these tiny components of our blood are far more complex than they seem.
Absolutely. And we're just getting started.
I can't wait to dive even deeper. But before we move on, there was one thing that really stuck with me from earlier.
Okay, what's that?
You mentioned that glycosylated hemoglobin, or HbA1c, is used to monitor diabetes. What's the connection there?
That's a great question. So HbA1c is basically a measure of how much glucose is attached to hemoglobin over time.
Okay, and why is that important for people with diabetes?
Well, for people with diabetes, their blood sugar levels are generally higher than normal.
Right.
So more glucose ends up sticking to their hemoglobin,
Oh, I see.
which means they have a higher HbA1c level. And by tracking those levels, doctors can get a sense of how well a person's diabetes is being managed.
So it's like a long-term view of blood sugar control.
Exactly. It's a really valuable tool.
That's incredibly helpful information. I'm sure many of our listeners who are living with diabetes or know someone who is will find that really insightful.
I hope so. It's just another example of how understanding these tiny details can have a big impact on our health and well-being.
Well, I'm definitely feeling a newfound respect for red blood cells and all their intricate functions. It's amazing how much we can learn from just one physiology handout.
It really speaks to the power of scientific inquiry and the vast amount of knowledge that's out there waiting to be uncovered.
And speaking of uncovering knowledge, we were talking about the importance of diet in erythropoiesis earlier. Are there specific foods that are particularly good for boosting red blood cell production?
That's a great question, and it's one that we could definitely delve into further.
Should we add that to our list for a future deep dive?
Absolutely. It's a topic that I think many people would find interesting and practical.
I'm already excited to explore that. But for now, let's shift our focus back to hemoglobin and its fascinating interactions. We've talked about oxygen binding and 2,3-DPG, but I'm curious about what happens when the iron in heme gets oxidized to ferric iron.
Ah, yes, the formation of methemoglobin.
Right. We touched on it briefly before, but I'd love to hear more about it. What exactly is methemoglobin and why is it important?
Methemoglobin is essentially a form of hemoglobin where the iron atom in the heme group has been oxidized from its ferrous state, Fe2+, to its ferric state, Fe3+.
Okay, and what's the significance of that change?
The problem with methemoglobin is that it can't bind to oxygen. Remember that ferrous iron, Fe2+, is crucial for oxygen binding. So when the iron gets oxidized to ferric iron, Fa3+, hemoglobin essentially loses its ability to transport oxygen effectively.
So it's like the iron loses its grip and oxygen can't hitch a ride anymore.
That's a great analogy. And as we mentioned earlier, a small amount of methemoglobin formation is normal. Our bodies have that handy enzyme system, NADH methemoglobin reductase, to convert methemoglobin back to regular hemoglobin and keep things in balance.
But what happens if that balance is disrupted?
Well, if methemoglobin levels get too high, it can lead to a condition called methemoglobinemia.
Methemoglobinemia.
Yeah. This can be caused by a variety of factors, including exposure to certain drugs, chemicals, or even some foods.
So it's like those external factors are overwhelming the body's natural defense mechanism.
Exactly. And the symptoms of methemoglobinemia can range from mild, like a bluish discoloration of the skin, cyanosis,
Oh, wow.
to more severe, like shortness of breath, headache, dizziness, and even seizures in severe cases.
That sounds pretty scary.
It can be, especially in severe cases. But thankfully, most cases of methemoglobinemia are treatable.
That's good to hear. But it definitely highlights the importance of understanding these different forms of hemoglobin and how they can impact our health.
Absolutely. It's a reminder that even seemingly small changes at the molecular level can have significant consequences for our overall well-being.
Well, I'm feeling a little overwhelmed by all this information, but in a good way. It's amazing how much complexity there is in something as seemingly simple as a red blood cell.
It really is. And we've only scratched the surface. There's still so much more to explore. And the more we learn, the more we realize how interconnected and beautifully orchestrated these biological processes are.
Well, I'm ready to keep exploring. What else can you tell us about these remarkable cells?
It's true. We tend to think of hemoglobin as just, this one thing.
Yeah.
But there are these variations that play specific roles throughout our lives.
Oh, wow.
Yeah.
Tell me about these different types of hemoglobin.
Okay, so the most common type, the one we see in adults, is hemoglobin A.
Hemoglobin A.
Right.
Right.
And there's a minor variant called hemoglobin A2.
Hemoglobin A2.
But things get really interesting when we go back to, fetal development.
Oh, okay. What's different about fetal hemoglobin?
So fetal hemoglobin, also known as hemoglobin F,
Okay.
it has a higher affinity for oxygen.
Higher affinity. What does that mean?
It means it can grab oxygen more readily from the mother's blood.
Oh.
Which is super important for development, in the womb.
That makes sense. The fetus needs all the oxygen it can get.
Exactly.
So it's like fetal hemoglobin is a super efficient oxygen magnet.
That's a great way to put it.
Okay, so we've got adult hemoglobin and fetal hemoglobin.
Right.
Are there any other types?
Well, there are even earlier forms
Oh, wow.
called embryonic hemoglobins.
Embryonic hemoglobins?
Yeah, showing just how, dynamic this molecule is throughout our development.
Wow, so it's like a whole timeline of different hemoglobins, each one fine-tuned for a specific purpose.
It's really amazing how our bodies adapt.
It really is. And it's incredible how much is happening that we're completely unaware of.
Right, like all these processes happening behind the scenes.
Yeah, exactly.
Speaking of which, remember that competition between oxygen and 2003-DPG
Yeah.
for binding sites on hemoglobin?
Oh, yeah, the party crasher analogy.
Exactly. So 2,3-D-TBG actually plays a crucial role in regulating oxygen release.
Okay, how does it do that?
Well, when 2,3-DPG binds to hemoglobin,
Mm-hmm.
it makes it easier for oxygen to detach and be delivered to the tissues.
Oh, so it's like it's helping to offload those oxygen molecules.
Exactly, right where they're needed.
Wow, so it's not just about how tightly oxygen binds, it's
Yeah.
also about how easily it lets go.
Exactly. It's all about balance.
And I'm guessing that balance is influenced by different factors?
Oh, absolutely. Like, think about exercise.
Okay.
Your muscles produce more 2,3-D-3-DPG.
Oh, so they can get more oxygen.
Exactly. It helps them keep going.
That's incredible. Our bodies are so smart.
They really are, constantly adjusting and fine-tuning.
So we've talked about situations where things are working properly.
Right.
But what about when the balance is disrupted? What can go wrong?
Well, we already mentioned carbon monoxide poisoning.
Right?
where carbon monoxide basically steals those binding sites. But there are other situations, too, where hemoglobin can be affected.
Like what?
Well, remember when we talked about methemoglobin, where the iron in heme gets oxidized?
Right, the one that can cause that bluish skin discoloration.
Exactly. So while some methemoglobin is normal, certain drugs and chemicals can accelerate that process.
Oh, wow.
Leading to something called acquired methemoglobinemia.
Acquired methemoglobinemia?
Yeah, it's a mouthful, I know.
So it's like those external factors are messing with the hemoglobin machinery.
Yeah, that's a good way to put it.
And luckily, our bodies have a system for dealing with that, right?
Yes. Remember the NADH methemoglobin reductase system?
Oh, yeah, the cleanup crew?
Exactly. It converts methemoglobin back to regular hemoglobin. But there are rare cases where people are born with a deficiency in that system.
Oh, no.
Yeah, it's called congenital methemoglobinemia.
Congenital methemoglobinemia.
That sounds serious.
It can be, because it disrupts the body's ability to transport oxygen.
Oh, wow.
But thankfully, it's very rare.
Well, it's a good reminder that even these tiny components of our bodies can have a big impact on our health.
Absolutely. It's all interconnected.
It really is. We've covered so much ground today. I feel like I have a whole new appreciation for these tiny cells that are constantly working to keep us alive.
Me too. It's amazing.
And we're not done yet, right?
No.
There's still more to discover.
Oh, yeah. There's always more to learn about red blood cells.
Well, I can't wait to dive even deeper. But before we move on, there was one thing that really stuck with me from earlier.
Okay, what's that?
You mentioned that glycosylated hemoglobin, or HbA1c, is used to monitor diabetes. I'm curious, what's the connection there?
So HbA1c is basically a measure of how much glucose is attached to hemoglobin over time. Okay, and why is that important for people with diabetes?
Well, people with diabetes have higher blood sugar levels than normal.
Right.
Which means more glucose sticks to their hemoglobin.
So they'll have a higher HbA1c level.
Exactly.
And what does that tell doctors?
It gives them a good idea of how well a person's diabetes is being managed over weeks or months.
Oh, so it's like a long-term snapshot of blood sugar control.
Exactly. Instead of just a finger prick test, which only shows you what's happening at that moment.
Right, right.
So it's a very valuable tool for monitoring diabetes.
That's really interesting. I'm sure a lot of our listeners will find that helpful.
I hope so.
Yeah.
It's all about understanding these little details that can make a big difference.
Well, I'm definitely feeling a newfound respect for red blood cells and all their functions.
It's amazing, isn't it?
It really is. And it's incredible how much we can learn from just one physiology handout.
Right. It really makes you appreciate the complexity of the human body.
It does. And speaking of complexity, we were talking about the importance of diet in erythropoiesis earlier. Are there specific foods that can help boost red blood cell production?
That's a great question, and it's definitely something we could explore further.
Should we add that to our list for a future deep dive?
Absolutely. I think a lot of people would be interested in that.
I'm already excited to learn more. But for now, let's shift our focus back to hemoglobin and its interactions. We've talked about oxygen binding and 2,3-DPG, but I'm curious about what happens when the iron in heme gets oxidized to ferric iron.
Oh, yes, the formation of methemoglobin.
Right. We touched on it briefly before, but I'd love to hear more about it. What exactly is methemoglobin and why is it important?
Methemoglobin is basically a form of hemoglobin where the iron atom in the heme group has been oxidized from its ferrous state, Fe2+, to its ferric state, Fe3+.
Okay, and what's the significance of that change?
Well, the problem with methemoglobin is that it can't bind to oxygen. Remember how that ferrous iron, Fe2+, is crucial for oxygen binding. Well, when the iron gets oxidized to ferric iron, Fe3+, hemoglobin can't transport oxygen effectively.
So it's like the iron loses its grip and oxygen can't hitch a ride anymore.
That's a great analogy. And as we said before, a small amount of methemoglobin is normal. Our bodies have that enzyme system, NADH methemoglobin reductase, to convert it back to regular hemoglobin.
Right, the cleanup crew.
Exactly. It keeps things balanced. But what happens if that balance is disrupted? Yeah, it's been quite a journey into these tiny cells.
It really has. I'm still amazed by how they managed to squeeze through those tiny capillaries.
It's pretty remarkable, right? That biconcave shape and that flexible membrane, it's all perfectly designed for that.
Yeah, it's like they're tiny contortionists just slipping through those tight spaces.
I like that analogy.
Yeah.
And remember, that shape also maximizes their surface area.
Oh, right, for gas exchange.
Exactly. They're incredibly efficient.
It's amazing how it all comes together, form and function, even at the microscopic level.
It really is.
Makes you wonder just how many red blood cells are out there constantly working away.
Millions upon millions. It's mind-boggling.
delivering oxygen, removing carbon dioxide, all while navigating this intricate network of blood vessels.
It's a symphony of activity happening inside us all the time.
Speaking of intricate processes, I'm still trying to wrap my head around the whole oxygenation versus oxidation thing.
Oh, right, yeah. It can be a bit confusing.
Can you remind me of the key difference again?
Sure, so when oxygen binds to the iron in heme, it's a reversible process called oxygenation.
Oxygenation?
Yeah, think of it like oxygen is just hitching a ride.
Okay.
It can easily detach when it reaches its destination.
So it doesn't permanently change the iron atom.
Exactly. The iron stays in its ferrous state, which is important for hemoglobin to keep doing its job.
And oxidation is when the iron changes.
Right. It loses electrons and becomes ferric iron.
That's what happens when methemoglobin forms.
You got it. And as we talked about, some methemoglobin is normal, but too much can cause problems.
Right, because then the hemoglobin can't carry oxygen as well.
Exactly. Luckily, our bodies have that enzyme system to convert it back.
The cleanup crew.
Yep. Always working hard to keep things in balance. Speaking of balance, we also talked about how red blood cells affect blood viscosity.
Right, how thick our blood is.
What happens when that's out of whack? Well, if blood viscosity is too high, it can strain the heart.
Oh, okay.
Potentially leading to cardiovascular problems. And if it's too low, you can have problems with clotting and wound healing.
So it's got to be just right.
Exactly, like Goldilocks in a porridge.
That's a great analogy. Well, I think we've covered a lot today.
We have. It's been a fascinating journey into these tiny cells.
From their structure to their function and all those intricate interactions, it's clear that red blood cells are so much more than we think.
They're essential for life.
Absolutely. And for anyone who wants to learn more about how to support their red blood cell health, we'll be doing a deep dive into specific foods in our next episode.
That's right. Stay tuned for that.
I can't wait to learn more, but for now, I think it's time to wrap things up.
Yeah, I think we've given our listeners plenty to think about.
We have. It's amazing how much complexity there is in something as seemingly simple as a red blood cell.
It really is a testament to the wonders of the human body.
Well said. And on that note, thanks for joining us for this deep dive into the fascinating world of red blood cells.
It's a pleasure.
Until next time, keep those red blood cells flowing.